Thanks everyone for joining us at the online live event to learn. I'm Mika, the host from AI Camp. So before we get started, just want to do a quick introduction on AI Camp. So we are a global online platform for developers, engineers, and data scientists to learn and practice AI technology, with the mission of making AI available to all developers. AI Camp have grown to over 70,000 tech engineers in the group, have hosted 300 local tech meetups, workshops, boot camps, large tech conferences, and live stream most of the tech talks globally. We have local study groups in the major cities in the US, a few cities in Europe, India, China, Australia. You can take a look at the website to see our upcoming tech talks, workshops, and crash courses that we offer. So starting next week, let's see. On February 12th to February 21st, on every Tuesday and Thursday at 4 to 6 p.m. Pacific time, we will have our online crash course on best practice for machine learning engineers. We present a set of best practice for software and system engineer that can be applied to machine learning application development. The course will cover a full gamut of required knowledge ranging from software tools, model selection and infrastructure to deployment and quality assurance process. And I don't know how many of you guys are based in the Bay Area. So if yes, please attend to our bootcamp on February 23rd, Machine Learning in Action. It will be a one-day training which will get you up to speed on machine learning technology. You will learn recognized problems that can be solved with machine learning, select the right technique for the problems, and more. And you will have 4 hands-on code labs as well. So I know that's lots of information to take away, but welcome back to our main topic today. So today we're excited to have Ali. Ali is currently a PhD candidate and at the University of Toronto working on the development of new machine learning models to predict cancer patient response to drugs. So during his study, he published research paper in high-impact scientific journals and international conference covering such fields as ensemble learning and unsupervised clustering. He have earned a Master of Mathematics degree from the University of Waterloo. So without further ado, let's welcome Ali to talk about dimension reduction today.
Thanks, Mika, for the introduction. So let me share
your slides. Let me unshare my slides now.
Okay. So do you have my slide?
Yes, I have your slide.
Perfect. Okay, hello everyone. I hope you're having a great morning, afternoon, or evening, wherever you are in the world. I'm Ali Madani. This is my full name if you want to find me in social media or Google me. And we're going to talk about dimensionality reduction. I wanted to first thank my friend and colleague Farnoosh, who helped me in preparing the material of this webinar. We are researchers in Princess Margaret Cancer Center in Toronto, as you see in this beautiful picture. And the focus of our work is development of machine learning models for predicting cancer patient response to drugs or predicting their survival. So this webinar will have 2 parts. The first part will be just more kind of introduction and why we care about dimensionality reduction, what are the approaches that we are going to talk about. And the second part will be a Colab notebook, or just you can open it as a Jupyter notebook I prepared for this webinar. And we're going to work on Python to see how we can implement those methodologies in Python for a specific data set. Whenever you are importing your data in Python or any other programming languages, you deal with data frames, or in mathematical notation, you may have matrices if you want to talk about the math behind it. And you have rows, which are data points, or it could be patient samples, images, or any other application that you want to work on. And you have columns, which are your features. Here, for example, is ID, address, ID, which is for this data points that we have, address, number of bed, number of baths, which is for real estate kind of analysis. We can have genes, which would be for genomic data analysis. And when we talk about dimensionality of the data, we talk about number of features that the data has. But the question is, what could be the range of number of features that we may have in a specific application? Image analysis or image classification, any other kind of analysis of images have been very hot area during the last few years. If we work on UCI ML handwritten digits data, which we're going to work on today, we can see 8 by 8 pixel images, 64 features. Each pixel is one feature in your model. So this is very maybe low dimensional kind of data. With respect to high resolution images that you may have more than 3 million features or pixels. But the question is that, do we need all those features for our analysis or not? There could be three main aspects of benefiting from dimensionality reduction. First one is for eliminating irrelevant features for a specific purpose in our analysis or reducing noise. Imagine you have two dimensions. Dimension 1 is feature 1, dimension 2 is feature 2, and you have two clusters of data points, red ones on the right and blue ones on the left. You agree that we need only dimension 1 or feature 1 to be able to separate these two clusters. So we can get rid of dimension 2 or feature 2 and reduce the dimensionality of data from 2 to 1 regarding our goal for separating these two clusters. The second important point is reducing time and memory in computation. When you work with bigger data frames, when you have more number of columns, the storage that you're going to use will be bigger, and also the computational time, the computational cost in your analysis will be huge. So we want to somehow use dimensionality reduction to have informative features, but smaller data frame. And the third benefit of dimensionality reduction, which was actually the reason behind some of the methodologies like t-SNE, is visualization. Imagine you have very high dimensional data, but you want to see if there is any kind of grouping between the data points. We cannot imagine more than 2D or 3D. Actually, if you want to work on your monitor or a PDF file or a piece of paper, we can easily discriminate points or understand the structure of the data only in 2D, and 3D will be even very hard. So these are the main three benefits of dimensionality reduction. Now let's imagine you have 5 original features and we want to implement dimensionality reduction. We can have two very general scenarios. First one is feature selection. So you're subsetting your features and going from feature 1, 2, 3, 4, 5 to new feature 1 and new feature 2, which are actually your original feature 2 and feature 4. So this is feature selection, which was similar to the example I had previously for dimension 1 and dimension 2, which we chose only dimension 1. But the other approach is feature extraction, in which we combine linearly or non-linearly some of the original features. Here, imagine you have feature 1, 2, 3, 4, 5. We are combining feature 2 and 3 to come up with new feature 1, and we are combining feature 4 and 5 to come up with new feature 2. So these are two general kind of approach for dimensionality reduction, for reducing the number of features. The approaches that we're going to talk about today are mainly in the category of feature extraction, because they combine features to come up with new dimensions. Let's have a real life example. The names are arbitrary and the scientific conclusion may not be perfect, but this is just an example. Imagine we have a friend Joe and another friend James, and one of them, Joe, is 170 pounds and 6 feet tall, and James is 150 pounds and 5 feet 3 inches tall. So the question is, if we talk about diabetes, we know that if we get fat and actually our weight will be higher, we may have higher risk of diabetes. But the question is that when we compare different people, do we just have to care about one of the features? No, because the answer to the question that is the risk of type 2 diabetes higher for Joe cannot be correct using only weight. For that, scientists, actually we all know about this feature, BMI, came up with this nonlinear combination of weight and height of the people. And then here we can see that James has higher BMI, which is 26.6. So based on the research, he may have higher risk of type 2 diabetes. Also, I'm not an expert in this area. You have to read papers about that. But you understand my point with this example, that we need to sometimes even combine features to understand the truth behind the data set and the structure of the data. We want to talk about three main and widely used approaches for dimensionality reduction today. The first one is principal component analysis, or PCA. Here is a beautiful review paper which discuss about the math behind dimensionality, math behind principal component analysis, and some of the examples for application of principal component analysis. There are also some derivations of principal component analysis that was discussed in this paper. But if we go back to the original papers that maybe discuss about this concept, or at least the eigenvalue, eigenvector kind of concept that is behind this methodology, we go back to 1901 and the paper that published by Carl Pearson. So we have a huge history for PCA. And around 10 years ago, there was another methodology which is now widely used, t-SNE, published in this concept, which I will cover later on. But for PCA to have a summary of how it works, imagine you have dimension 1, feature 1, and dimension 2, feature 2. PCA tries to combine linearly these features to come up with new dimensions or features that explain the variance of the data as much as possible. Based on this concept, PCA will give us something like this. So the first dimension will try to explain the variance or scatterness of the data as much as possible. And after deducing the variance explained by this dimension, then PCA tries to explain the rest of the variance as much as possible using the next dimension or next principal component. As we can see here, the new dimension will be the linear combination of the original features, but the coordinates are different. And at the end, instead of having feature 1 to feature P, we will have principal component 1 to principal component K. Sorry about that. And each one of the data points that we originally had, for example, it could be images, will have a score for each one of the principal components. So our data frame or matrix in mathematical notation will be samples or data records or data points and principal components in columns. And number of principal components less than or equal to minimum of number of the original features that we have and number of data points. And this is actually could be number of data points minus 1 if we work with centralized, column centralized data. For example, if you have 2 points, you only need 1 line or 1 dimension to describe all the data because you have only 2 points. t-SNE was another methodology published in 2008 by van der Maten and legendary machine learning scientist, Dr. Hinton. And it was very interesting for me that actually Dr. Bengio was the editor of this paper. These two people are amazing machine learning scientists and actually the legends in the field of computer science and machine learning. And there's an amazing GitHub page by van der Maten, which you can find the description of t-SNE, the code, and some examples. I'm going to answer if you have any question at the end of the presentation, then we go for the Python code. Let's have a summary overview of what exactly t-SNE does. So t-SNE tries to reduce the dimensionality of the data, for example, from p-dimensional space to 2D space. And in this case, if you have, for example, here 2 clusters of points similar to what we had in the previous example, we can see that t-SNE tries to preserve these groupings. So that's the point. t-SNE cares about neighborhood. It tries to preserve local structure of the data and bring the neighboring points from the high dimensional space to a neighboring embedding in the low dimensional space. And we can see that the clusters could be preserved while we cannot discuss about large distances, and it is very important point. When we have t-SNE, when we use t-SNE on our data, we cannot talk about distances between the clusters or large distances in our plot. To somehow resolve that issue, not exactly for solving only that issue, Tufts Institute researchers developed another methodology in 2018, last year, called UMAP, or Uniform Manifold Approximation and Projection. Here is again another amazing GitHub repository by the first author, Leland McInnes, that you can find different code example and just a description of how it works. But in UMAP, the important point is that in addition to trying to preserve the local structure, UMAP also tries to preserve global structure of data as much as possible. So we can discuss about distances between the groups and clusters. We will see the examples in the Python code. It was just an overview of the methodologies and intuition behind dimensionality reduction. So we will cover PCA, t-SNE, and UMAP in our Python code, and then discuss how we have to interpret them, or what are the assumptions behind them, or what are the important parameters in the model. So we will discuss about the different parameters in the model as well. So let me see if you have. So I will go through the Colab notebook, and I will actually give you the link. So let's go to the Colab notebook. So here is just a Colab notebook I prepared. You can download it and work as a Jupyter notebook. Here is the link. So you can use this link. I will wait for a couple of seconds.
Or Ali, are you able
Yes.
to put the
Do you
link?
want me to copy it in the chat?
Yeah.
Okay. Okay.
Easier.
Yeah, I agree. So it's better. Okay. So this is read-only Colab notebook, but you can use it. You cannot edit the same file, but you can download it and use it yourself and edit it later on. I will also push this code to my GitHub repo later on if you want to work on it.
Okay, sure. Thank you, Ali.
No worries.
Okay, and just one quick question.
Sure.
On slide 13, you describe the feature construction, but they think it seems like it's feature construction, not feature extraction. So they want to know if you can clarify that one.
So I agree. It's a matter of methodology. So sorry, terminology. Some people call them extraction or construction, but technically we are constructing new features, or we are extracting by linear combination new features. So it's a matter of terminology. But in the broad kind of perspective, we have two main categories of feature selection, which is subsetting of the features, and feature extraction or feature construction. Yeah.
Okay.
So any other question?
No.
Okay, so it's about the link. Okay, perfect. Okay, so if you are not that much familiar with Python, I will try to describe a little bit what's going on in the Python code, but the main goal is to understand the dimensionality reduction approaches. So we are importing different packages we need. NumPy is the fundamental package for scientific computing. Matplotlib, we need them for plotting. And scikit-learn is backbone of machine learning in Python. UMAP, because it was a recent methodology, has its own library, but it is compatible with scikit-learn. So the goal of this Colab notebook is working with different dimensionality reduction approaches. And the data that we are going to use is UCI ML digit image data that I described actually in my presentation. Those are 8 by 8 or 64 pixel images, and there are around 1,700 images. It is smaller than MNIST, and it will be easier to work with for webinar. First of all, we are going to import the data from scikit-learn. We import the data set. We load the digits. And there are two parts in this object. We have the data, which is the data frame. Rows will be the images, so the data points, and columns will be the pixels or features. And target is actually the label to say that if this is an image of 0 or this is an image of 1 or so on. And the range of the pixels is between 0 and 16. So here it's 0. I can just uncomment it and just we can see that the maximum point is 16. And here is just some example of the images that we are dealing with in this data set. So we are building some subplots. Each one of them is a subplot, and we are plotting 10 of the digit image data that we have. Okay, so let's start working on PCA. As I mentioned, the goal of PCA is to reduce the high dimensional data to low dimensional data, but preserve the global structure of the data and try to explain as much variance of the data as possible from the first component to the last component. So the first principal component tries to explain as much variance as possible, and then the second one, and then the third one, and so on. Here, if we imagine this synthetic data, so I generated this synthetic data here. We have dimension 1 and dimension 2. And these are points in 2 lines, which are somehow parallel. Just I added some random Gaussian noise for each one of the points. And now we want to see how PCA tries to reduce the dimensionality. If we go back to the presentation, we expect that the first principal component will be some kind of diagonal line here, which makes sense because it will describe the maximum variance possible. So as I mentioned, scikit-learn is the backbone of machine learning in Python. From scikit-learn, we are importing the composition module, and PCA is a class in the composition module. So this part is not something maybe very important for the dimensionality reduction. I'm just building the data frame that we can see the dimensions here, the shape here, with the points that I generated here. So we have these points. I'm just generating a data frame using the 2 features. And now we want to use this data frame in our PCA analysis. We only need 2 components because we just want to visualize it. We don't have any other goal for this example. And using scatter from PLT, PLT was the abbreviation for matplotlib.pyplot. We can see that the first component is almost a diagonal line. It's something like this in the original data. And the second dimension, similar to the example I had in the presentation, is somehow perpendicular to the first principal component or first dimension. So it agrees what we described. Sorry, this A should be capital. So we understood how PCA works on some synthetic data. Now we want to see how we can use it to separate maybe the digit image data that we have. So again, to review from scikit-learn, we are importing the composition module. And in the decomposition module, we have PCA class. Number of components, we just want to visualize it for now. So 2 components, the first 2 principal components we want to use. And we are feeding the model using the data frame that we have. So the columns are pixels, which are our features, and the resulting shape, the shape of the resulting data frame is 1797 because we have 1797 data points, and the number of new features is 2 because we selected to have 2 components. Here is just a plotting function I defined to make some beautiful plots for the rest of the analysis. And if you have any question, I can answer at the end. So I'm using this plotting function to scatter plot this data frame, the resulting principal components. And as we can see, there is not much of a separation between different digits. So first of all, definitely it preserved the global structure of the data. If you consider all of the principal components, these are only the first 2, but it wouldn't separate the cluster of the groups of digits as we expected. Maybe some of you say that, okay, so we have zeros here, we have other numbers close to each other, but you have to remember that we don't have these colors. PCA is unsupervised. What we have to look at is something like this, which is exactly the same without any coloring. So when we look at this plot, there is no structure. So we don't see any separation between different groups. We just add the colors to just investigate or validate some of our observation that we may had before. One of the reasons could be because of the variance explained by the first two components. Here, I'm doing the same principal component analysis, and instead of 2 components, I'm selecting 50 components. In the principal component object that I will get, there will be explained variance ratio. And this explained variance ratio will be for the first component first, and then second, and so on, which I printed here. If we plot the cumulative summation of the variance, the fraction of the variance explained by each one of the components, we can see that it increases and go up to 1. So when we have all of the components, it tries to explain all of the variance. So the fraction should be 1. But if we look at the first component, it only explained less than 15% of the variance of the data. The second, less than 14%. And in total, it's less than 28% of the variance of the data. And not necessarily 28% could be enough to understand the structure of the data points. If you want to have a rule of thumb of how many principal components we may need to keep, first of all, you can keep all of the principal components. Sometimes you prefer to actually keep the first few, but the rule of thumb is 70% of the variance explained. If you keep the first, for example, here, the first 10 principal components that explain more than 70% of the total variance, maybe it would be a good choice. So let me see if you have any question before going to t-SNE. This was an overview of principal component analysis using the digit data, but I will compare it with t-SNE and UMAP during the talk. So, yes. PCA. Okay, great question. Before just I move to t-SNE, what is the difference? The question is, what is the difference between PCA and ICA? In a very general overview, PCA cares about correlation between the features, between the new features. So we will have uncorrelated features, while ICA try to reduce the dimensionality of the data and come up with new features which are independent. And there are, for the interpretation of ICA, if you want to work on ICA, the important thing is that ICA will not give you the new features which explain the variance in order. So you do not see this kind of ordering of the explained variance by different features, different new features. So I will go through more details at the end if you have time, but this was the general difference between ICA and PCA. So for the original data from PCA in general, so if you have all the principal components, you can identify the original data most of the time, because it's somehow a mapping scenario. But let me just go through the rest of the presentation and I will answer questions, because you may have actually more questions from t-SNE and UMAP because they're a little bit more complicated and I want to have enough time to answer the questions. So t-SNE, there's a difference between t-SNE, UMAP, and PCA. And that is about the global structure versus local structure. PCA, as I mentioned, try to preserve the global structure of the data, try to explain the variance in the data. But t-SNE tries to preserve local structure, as we discussed. If there is any kind of neighboring or grouping in the high dimensional space, we expect to see t-SNE tries to give us such kind of embedding that preserves that local structure or that neighboring in the new low dimensional space. Here, similar to PCA, it's fairly easy. From scikit-learn, you are importing manifold module. t-SNE is a function, is a class in the manifold module. we want to work with two components, and there are rest of the parameters of the function that I'm going to work, that I will describe later on. And we are feeding our synthetic data. If you remember, we had two lines, and we see that the resulting embeddings is not similar to PCA, but we have the separation of the lines. So just to describe this important parameter, these ones I will describe later on, but this is initialization by PCA. t-SNE is not a very fast methodology. So when you use PCA to reduce the high dimensional data first to a low dimensional principal component data frame, then you use t-SNE, it will be much faster. And this is exactly what it does. So it reduces, for example, if you have 1,000 features, it uses PCA, reduce the dimension of the data, then it applies t-SNE on top of that to get the two-dimensional t-SNE plot. Now let's use t-SNE on our digit image data. So from scikit-learn manifold module t-SNE class, again, two components, initialization by PCA, and I will describe these a little bit later. And now what we see is the separation of the digits from each other, the groups of digits, which we expected. We expected to see these kind of clustering in the high dimensional space, and t-SNE could preserve it in the low dimensional space or 2D space that we have now. But the important point, as I mentioned, is that we cannot discuss about the distances that we have between these clusters. But we can say that these points were actually grouped together or were nearby in the high dimensional space. So before going to the parameters, let me check if there are other questions. So first, destroy the locality conservation of t-SNE. Not necessarily. So, wow, we have a very one paragraph of comments, so I will answer that later. But PCA, as I mentioned, just keep the global structure of the data. And if you have all of the principal components, not necessarily you will lose the locality. We lost locality before in PCA because we had only the first two principal components. And we cannot use more than two for visualization. So that's a trick, actually. So if you could imagine, and if you could somehow interpret 50-dimensional space, and I use the 50-dimensional space, we could see the clusters, maybe. So it depends on the data as well. Now let's work on the parameters. So perplexity is maybe the most important parameter. Number of iterations is similar to any other optimization process, or if you're working numerical analysis, it's just a matter of convergence of the methodology of an optimization process. But perplexity determines the structure of the data in your two-dimensional space. So what perplexity means is that if you have one data point and you're trying to bring that one data point to the low dimensional space from high dimensional space, how many nearby points will affect the mapping of that point? Is not similar to K and N or so. There will be a distribution around each one of the points. And when we increase perplexity, we are increasing the effect of distant points on mapping of that target point. So what we expect is that, so if I go back here in this beautiful plot, we had perplexity of 30. If I use higher perplexities like 200, what we see here is that we are losing the structure here. Okay, so it is very important because for every point in the high dimensional space, now it cares about a lot of distant points in other clusters or groups. So it loses the specificity of neighborhood in each one of the clusters, which is very important. And if you use lower, I hope I don't need to run the whole notebook again. No, I was connected before. Perfect. We see very interesting and misleading clustering. So we have these kind of pinchy shaped clusters, And this is the result of this low perplexity. Although it is in the suggested range, it's better to stick to the middle of the range, usually. So the reason for that is that imagine you have all these points, similar to here. In the high dimensional space, if we use very low perplexity, it means that only the nearby points around that target point